bring that exact point here to some of these open data environments. This is the collagen gene that Taylor mentioned is highly expressed by lines that are going off target to mesendodermal lineages. And so this is a profile of data sets in NeMO Analytics where we've collected all the data that Silke just talked about. These are the first 6 set of lines in the 3 differentiation protocols, the knockout, the second set of lines, the next gen data. But what we can see already here, the expression of that when off target, meaning off target for dual SMAD inhibition, this collagen gene is being turned on spontaneously in line I4 in pluripotency. This turns out to be the line that Silke showed right after Taylor showed this gene that is not responsive to multiple doses up to 10 microgram or micromolar, by the way, which is right in the middle of the low dose of Taylor's. So that it kind of brings together a lot of what we've seen today. In pluripotency, before these cells are going anywhere, the cell line that we know does not efficiently hit the neural induction when it gets dual SMAD inhibition is already showing the collagen that Taylor found as a reporter for that off-target mesodermal lineage. And here you can see it come on in all the lines when they're hit with BMP4. So I want to kind of make the conceptual point here, but also the practical point that I will send to the email group and the Slack channel links to all these data sets so you can see them. And as you can see, it's quite simple. You type in a gene name and it shows you that gene in all the data sets. The collagen in the next gen data is, there's a couple high expressors here in the neural end, which is interesting or surprising, but most of it is down here going the other way. And this is how that data was expressed in the other figure. Here, instead of PC1 on this axis, it's the gene that we're requesting. And to show kind of the flip side of this, if we now look at a neural related gene, this is FESF2.
But Carlo, let me say, what you were saying is it's not PC1, but it is still, they're all ranked by PC1.
That is correct.
Right.
So the vertical axis is the gene, but they're still ranked.
Collagen 1A2 has nothing to do with the major PC, except perhaps down here at very high PC1 levels, there seems to be a class of humans. These could be your outliers. We would know unless we went and did some more thinking and do an experiment here. But it illustrates the point Carlo's making, I think, very nicely about how one can use these data and these approaches.
So this would be 5 lines from this one individual, all of which are relatively high in the retinoic acid expression, as you can see in blue, and pretty high in this collagen.
Carlo, my point, as I keep playing devil's advocate here, were all of those lines made by the same method? By that,
for sure they don't encompass the variation that you're looking for.
Yes,
for
exactly.
Sure. And
you know, sorry, Taylor.
my 2 cents would be those lines that had the COL1A2 issue, we were able to overcome that by growing them in a better media with the FJF2 disc, right? So I would like to know how these were grown, right? Or was this Caesar?
There's no disc in the Ehor lemniscus data for sure.
Yeah.
And we've
actually, if you guys have the single cell sequencing data or any sequencing data, instead of having a discussion about it, we could just look.
Yeah.
Because you talked about 100 cells, 100 individuals right at the beginning.
Yeah. So
didn't you?
Hundreds
of lines.
Yeah, those hundreds of lines. We have a project with Dan Gashwin's group, so they've been doing a lot of omics on all those lines. So we have single cell and RNA-seq, and
wow.
So we can look. Yeah.
Great.
Those lines were made in different places with different methods. So we'll see.
Interesting to see. Yeah. Are there replicate lines from different individuals included in the collection as well?
Yes. Yeah.
Cool.
Callie, you're obsessed with this. It's interesting. I want to make a comment to you, Sally, about the whole field. So there may indeed be different kinds of state that you can stabilize and stabilize by different growth conditions. I mean, one of the really surprising things to me when we started out in this kind of area, how many of these early cell lines, these early cell states, you can grow as lines at all, right? And that suggests that what you're saying is actually true, that there are metastable kind of proliferative states that you can support in culture in a self-renewing condition. It's extremely interesting, actually.
Exactly. I guess that's our feeling right now. And we want to do the experiment that we rate, right? We want to see for a single donor how much of this is really robust across the parameters that people use. You know, we're obsessed about it because we want to make these models as consistent, but we also want to make them, as everyone here does, reveal differences between individuals, and those individuals, you know, understanding, for example, their disease susceptibility. It may be in the signatures that Suki showed us today. I think it's tremendously exciting, and we haven't paid attention as a field. A lot of protocols we find when we analyze them, let's say for Cortex, they were developed over maybe using 3 lines or a small number of lines. And then when you try to apply it across many lines, you just run into problems. So we're trying to address this as a practical feature. But what this presentation raises is that it's really telling us so much more about IPS states and donors as well, potentially. It's
in
great.
her response to you earlier, Sally, Suki pointed out that the genes that are responsible for the identity are different. It's so I want to just to leave you with a strong impression that there's something real going on here. It's not just some sort of noise, right? Which is technical and should be ignored, right? Then she pointed out that they're more rapidly evolving. They involve all kinds of zinc finger genes. It takes you into Didier Trono land, right? And into a very interesting kind of space genetically about what identity is, right? And how identity seems to evolve in parallel with a kind of Waddington type control system. So we're all, it's like we're all versions of each other. If you see what I mean, if that's not too
That's very sweet. Yeah, I think it would be really fascinating to hammer in those different ways of making the cells. And of course, I know that you'll be interested in ancestry, which is really important to be able to look at with this approach. You know, different genetic ancestry.
So if you guys are interested, we can do a little more data surfing if you can hang out, and then when I send you these links, hopefully they can be useful to you. And if after this session you want more guidance on how to use these or how to get your data in here, please do reach out. We're very interested in both of those. So we just looked at the collagen gene. This is sort of the flip side. FESF2 is, in a lot of us growing cortical organoids, think about it as a master regulator of cortical fate, but it is active much before there's a cortex as well. You can see here in 2 days after dual SMAD, it starts to come on, especially in the line SAO1 that does it most efficiently. In the next gen data, where we see the population of humans, the FESF2 is really highly expressed in the neural biased end of the cloud here. So that's interesting to see. Thanks, Sally. And here it is expressed another way. This is that area in the middle of those 100 individuals, 300 lines that have high retinoic acid. There's high retinoic acid at the end as well. So these are actually 2 different sort of cassettes of retinoic acid response, one that's involved in neural differentiation and the other in mesendodermal differentiation. And we can see that differently across this map. In the mouse embryo, I think a lot of you are familiar with this data set, Pijuan Sala. the brain is developing right about here, and you can see FESF2 coming on, and specifically this is a subset of this where the epiblast is here and the nervous system is emerging right here. There's a very specific slice of actually dorsal and anterior fates emerging there, or actually dorsal caudal a little bit. I think Nicola could refine that. It's also seen in the monkey gastrula here as these lineages are emerging. And so just with 2 genes, you can kind of survey all these data sets. The other thing we can do quite nicely, in addition to looking at individual genes across all these data sets to understand all the points we've been talking about today, we have engineered into this site a way not only to type in a gene, but to type in a complex combination of genes. So here, this is the same bunch of data sets that we'll page through, but instead of having a gene here, we've loaded in Sulky's 22 patterns. And pattern 13 happens to be the H9 identity, or the transcriptional signature that's specific to that line. So in all 3 conditions, you can see this purple dot here is H9. It's expressed, and this is a combination of many hundreds of gene weights, if not thousands, across the genome. So you might have to do some digging to understand what's under the hood here. But the H9 shows up not only in our data, but if I pass these developmental data sets down to the bottom here, these are other data sets that include H9. So this is DNA methylation data. So you can see the genes at which H9 is highly expressing genes, it actually has lower methylation levels, consistent with a TSS or promotal proximal gene methylation that decreases expression. These are both pluripotency and embroid body differentiation of H9 here called WA9 among many other lines. You can see here the H9 signature is very stable. And these other 3 data sets underneath with the black and green are different data sets across the planet and across the years. These are 2014, 15, and 16 bulk RNA sequencing of 5 or 6 lines in each case, one of which was H9. So the fact that everyone is using this line gives us an sort of an interesting way to validate this, that in everybody's hands at every time in history in any lab, if you're growing H9, it is humming this transcriptional signature. Again, talking about how stable or unstable these lines are. So in another, so now if we just click on a different pattern here, this is pattern 12. So this is one of the dynamic patterns, highly induced in SAO1, to a lesser extent in UC06. We can see that that pattern in our 6 lines corresponds to the neural end of the broader population here in the next-gen data. And then when we go to the mouse gastrula, we can see it here. This is the presumptive brain in the Pijuan Sala data right around, what is this, P8 or E8.5. And so again, this is really, the idea here is to get data sets together, even at the gene level, that are relevant to our interests. And so you can look across them, because most data sets, that there's definitely a data deposition somewhere. You can go get the raw data. There's usually a website where you can look at one data set, but looking at them together is really something that we do a lot of here in NeMO that's not easy elsewhere. If there are any other questions, we can go over some. I have lots of other examples, but we're almost at a half hour over. And like I said, if you want to get your data in this system or want more information on how to use it, reach out to me and I'll be sending all these links so that you can do this same surfing we just did on your own. Is there anything anybody else wants to see that we saw today that we could bring up right now?
TGF-beta receptor 1, ALK5, if you've got it, if you got the time.
I already had it up.
Okay.
So yeah, so this is interesting. I don't know where you'd like to start looking, but clearly in pluripotency, there's differing levels. It looks like our I13 line, it has the lowest. I4 is the NSB not so responsive, and it's showing at least at day 6 the highest levels. Surprisingly, SAO1 is also here among the higher ones, but it is very responsive to NSB. So at least the RNA level here we wouldn't have a great predictor of what we've seen in the NSB responses, but we could look at some of these other data sets. It's pretty flat in this the second set of 6 lines. There's a couple that are low.
Mm-hmm.
Oh, this is interesting. So in the next gen data, it's not really biased, maybe biased toward this end here, the neural end, but really where it's low is this kind of middle swirl. And if you remember from Soki's last panel, that swirl is kind of obviously in between the neuroectodermal and mesodermal, and you can really see both SOX3 and FOXA2 are both expressed in that population. So it's really kind of the cells that are on the teetering point that haven't really slipped toward bias in one or the other of those lineages.
And those days, the 2, 3, 2, 4, 6 for the pluripotency, those are like days in culture. So it's like less confluent, more confluent.
That's exactly right. So that's days from passage.
Days from
Yeah, so that's something that really allowed Soki, and Soki jump in here with details, but it's something people don't do, right? If there's a pluripotency condition in a paper, it's all from the same time point, and it's a single time point, probably close to confluent, so they get a lot of RNA. But the course of passage to confluence really revealed the structure that Soki was talking about. These cells are their response to morphogens are highly variable across that passage to confluence time. And the knockout really depended on that as well. Because if you look at BMP responses right when they're passaged versus 2 or 3 or 4 days later, that changes entirely. So yes, that is 2, 4, and 6 days of pluripotency.
And sorry, what media is this in? Is this M-teaser? Yeah.
Yes, same teacher.
Okay.
Right.
Cool. Thanks.
It's very cool to see how intertwined your efforts are, and how interesting this is going to get as we get more lines, more replicates, and more humans to look at.
Yeah.
Tyler,
We're working.
can I ask? Is Tyler still here? Yeah. Can I ask you a question, which occurred to me earlier, but now's a chance maybe when we're winding down to ask it. So when you listened to the talk last week from the Harvard group about variation early, do you imagine that by the manipulations you are doing, you can actually stabilize the system in a way that would suit their needs?
Yeah. It was a very great talk last week. And I had a similar thought. One way they could go about that is do a different protocol and see if that diversity holds true. Is it really diversity from the line itself, or is it just the line behaving differently in the protocol, right? Is it a phenotype, or is it a technical issue with the protocol? Yeah, I think that's the problem we all face.
Yeah, if all their lines were coming from, say, a flat FGF2 level like you've done in your system, would a lot of those proliferative differences disappear or not?
Yeah. And what we see is that helped a few lines, but we had to tinker with the SB to kind of bring in the other lines. And there's probably still other, parameters you have to tweak. I would say definitely those like earliest stages in the protocol and the iPSL cells itself. I think those are really going to be the best way to sort of work on this. And yeah, we're fortunate to have these isogenic sets in our studies, but not every disease has those kind of tools that we can use.
Thank you.
Thanks, everyone. I appreciate sticking around so long. We've gone about half hour past the talks, and that discussion was really cool. I will share the links for data, and please do reach out if you're interested in more.
Thank you.
See you, everyone.
All right, thanks.
Bye. Thank you.